Hello everyone, in this video I'm going to be talking through the first part of protein synthesis that we call transcription. So there's two videos really to this kind of topic, this first on transcription and the second one on translation. Now DNA directs protein synthesis in the cell, and the sequence of bases in a gene directs the sequence of amino acids that you get in a polypeptide. Now in doing that, two major reactions are involved, and as I said, this first part is all about transcription. Now in this process, so in this first reaction we're going to talk about, a copy of a gene is made in the nucleus that carries the information needed to make a polypeptide in the cytoplasm in the form of what's called an RNA molecule. Now that RNA molecule is called messenger RNA or little mRNA. So what I'm going to do is talk through this whole process and explain what is actually going on in transcription. Now for this, we're going to imagine that this whole screen is essentially our nucleus in a way. And I'm just going to draw, if I draw this in black just to make it stand out, I'm just going to draw a portion of what would be our nuclear envelope around the outside. So if we imagine the whole of the almost left side of the screen is within our nucleus, what I've drawn here is the nuclear envelope. If we just label these parts so that we're clear. So this is meant to represent our nuclear envelope, and so these gaps that we're seeing here, these would be the nuclear pores. Now the reason I'm putting these in place is because the messenger RNA that we produce essentially will leave the nucleus through one of those pores. So what we want to do is start with a piece of DNA. I'm just going to draw a strand of DNA. Now you'll notice that in this central portion I have the strands almost split open in a way. It's as if you imagine a hand at the top of the screen and a hand at the bottom pulling the strands apart and exposing some of the bases. That's actually what happens. So what we have is our DNA strand, and if we just put some bases in place, if we imagine just for these side parts, I'll just draw some generic typical lines that people would recognise as a DNA structure. Then if we just put a few of the bases in place, so let's just put a few letters here. Now clearly this is not to scale, but we're going to say these are, let's just say T, A, C, G, G, A. We're just making up letters for the sake of it. Now the complementary bases, therefore, would be A, T, G, C, C, T. So all we've done is put in place there a piece of DNA whereby some of the bases have become exposed. Now what we need is the exposure of those bases, because you can't just take a piece of DNA out of a cell. So we use this template, if you like, to create a piece of messenger RNA that we're going to use as a sort of preliminary code in producing our protein. So we've exposed it, we've got this open piece of DNA. Now RNA polymerase is an enzyme, ends in ASC, so RNA polymerase binds to a site of the DNA called the promoter region, and this determines where transcription starts and which strand is transcribed. So what I'm going to do is just essentially put in place this, and I'm going to draw it a bit like a big blob here on the side, and we'll just shade this in green, because many diagrams actually show it, especially in textbooks, as this big structure that's going to work its way down the strand of DNA. So we'll just put that in place there, and we'll label this. This thing here is our RNA polymerase enzyme. So it binds to what's called the promoter region. So we're going to almost say, if I draw it in red here, if I put a little star, we're going to say that's our promoter region. Now what the RNA polymerase does is unwind the double helix to reveal the two DNA strands. So I've already kind of jumped ahead a little bit there and opened up this DNA and exposed the strands, but that's the job of this RNA polymerase. So it binds to a promoter region and it unwinds the double helix at the point at which is very specific, exposing certain bases that we need, the bases that we're going to use to produce a very specific protein. Now what that does is expose two strands of the DNA. We've got a sense strand and an antisense strand. Now the sense strand, if I just label this on the diagram, the sense strand will have the same sequence as the messenger RNA that we will produce. So the sense strand is this one at the top, and I'll explain in more detail what I mean by this in a moment. So this is going to be our sense strand, and the antisense strand has a sequence complementary to the messenger RNA, and it acts as the template strand, which is this one here. So this is our antisense strand. So, once we've got RNA polymerase binding and the exposure of these DNA strands, the sense and the antisense strand, free RNA nucleotides in the nucleus pair with complementary bases on the antisense template strand of DNA. Now, on a separate video, I've talked about the differences between DNA and RNA. Now, RNA is made up of 4 bases, but unlike DNA, where we have A, T, C, and G, adenine, thymine, cytosine, and guanine, we have adenine, cytosine, guanine, and uracil. So instead of the T, we have a U. So where we've got a T on the antisense strand, that would essentially, or rather where we have an A on the antisense strand, a complementary base pair of that would be a U as opposed to T in RNA terms. So what we end up with is something that looks a little bit like this. And if I draw this in, a sort of pink colour just to make it clear what we're talking about here. Or I'll draw this, in fact, in a darker green, just to really stand out. So, what you find is that RNA nucleotides start to bind to this exposed antisense strand, and clearly this would just go on here, etc., and we'd continue it along here. I've only drawn 6 bases on the antisense strand, but clearly there would be many, many more. So what we're getting is free RNA nucleotides within the nucleus. So if we just I'll just draw a few here almost. C, let's draw a G, let's draw an A over here. What these are just free RNA nucleotides, and they just move and just come and line up with their complementary bases. So where there's a free G, for example, here, that will move up and come and join by complementary base pairing to where there's a C on the antisense strand. So you can see you're almost like building a messenger RNA strand on top of the exposed antisense strand. And this here, if we just draw a little arrow here, this is what we call the direction of transcription, or downstream. So we'll just make a note of that. This is what we call downstream, and it refers to direction of transcription. Now, phosphates get released providing energy in the form of ATP, allowing subsequent nucleotides to join. So that's what triggers these nucleotides joining. RNA polymerase forms covalent bonds between the nucleotides to form a messenger RNA strand. So this is a strand, you can see these green RNA nucleotides, they're covalently bonded, thanks to the RNA polymerase. And we call this a messenger molecule because it's going to carry our message if you like. So what we're producing ultimately is an mRNA strand. So that's the key thing here. What we do is we produce an a small m RNA strand. And that stands for messenger RNA. It's this green strand that I've just been drawing in. Now, RNA polymerase moves along the DNA molecule. It unwinds the double helix as it goes, and it extends the transcript. Behind it, the newly formed RNA transcript separates from the DNA template, and the DNA double helix reforms. So essentially, as this RNA polymerase works its way down this DNA molecule, where I just put a number 1 here, these RNA nucleotides join to their complementary base pairs, and where I put a number 2, these RNA nucleotides essentially start to detach from their base pairs. So we're almost pulling off it. So if you imagine this green strip, we're peeling it away and allowing the DNA strand to essentially coil up again. Now, when RNA polymerase reaches a termination sequence, or what we call a stop codon, it detaches from the DNA and releases the newly formed RNA. So if I continue this, if I just put a little arrow from here, essentially, just to continue this part of the story. So we've produced a messenger RNA strand. We reach a stop, what's called the stop codon, and RNA polymerase detaches. Now, when that RNA polymerase detaches, as I said, this RNA strand, this messenger RNA strand, becomes free. So what we end up with, ultimately, within our nucleus, is this RNA strand. We'll just write the letters in that we've got here. So we've got an A, U, G, C, C, and a U. So all we've done is just detach this strand and let it become free. And as I said at the start of the story, once we've released that mRNA strand, that messenger RNA, that strand is then able to exit the nucleus via a nuclear pore and passes into the cytoplasm. Now it's there where that messenger RNA molecule will then move towards a ribosome for part 2 of protein synthesis, what we call translation. Now, just to say as a sort of added extra in this section, once we have this messenger RNA molecule, there are actually parts of it that are non-coding. We do have some non-coding DNA, and some of that non-coding DNA needs to be removed. Now, eukaryotic genes contain what are called introns and exons, and only the exons code for protein sequences. So the introns we can actually remove from the RNA to form what's called mature messenger RNA, and that gets spliced together in a process called splicing. So what we actually have is our DNA. Ultimately, we use that as a template to form an RNA transcript, and then we use enzymes called ribozymes and proteins within the nucleus to help us remove the introns and splice together the exons. So what we end up with is a messenger RNA purely of coding RNA, essentially. Now, after further modification, the mature messenger RNA, again, that's when it travels to the cytoplasm, where it can be translated. So that's just a little extra part at the end there. Okay, hope all that helps.